So, hemoglobin doesn't unload the same amount of oxygen to all tissues. So, the utilization coefficient is often more than 22%. Each tissue has different metabolic needs and different states of activity. So, there are factors that will adjust the rate of oxygen unloading. First of all, the ambient partial pressure of oxygen. Since active tissues consume oxygen rapidly, the partial pressure in those tissues is sort of low. Thus, there's more of a pressure difference between the partial pressure of oxygen in the blood than there is at the tissues. So we see a faster release of oxygen from the hemoglobin. Temperature has an effect. Active tissue has a higher temperature, and that increased temperature will promote oxygen unloading. When temperature rises, the oxyhemoglobin curve shifts to the right. So at 10 degrees C, it's more on the left. At 43, it's more on the right. 38 degrees C is about normal body temperature. So the elevated temperature essentially promotes oxygen unloading by shifting the curve down and to the right. The Bohr effect has to do with pH. Active tissue has a higher concentration of carbon dioxide, as we've seen that bicarbonate buffering system generates more hydrogen ions. The more hydrogen ions will lower the pH of the blood. As the pH of the blood is lowered, the oxygen dissociation curve shifts again down and to the right. You can see here, at a slightly lower pH, oxygen dissociates more rapidly. This effect is much less pronounced at high partial pressures of oxygen, so we don't see so much Bohr shift occurring in the lungs. In the systemic capillaries, however, the partial pressure of oxygen is fairly low, and we see a much more pronounced effect. So that is, a low pH or high acidity in the tissues promotes unloading of carbon dioxide. A final effect is the Biz Phospholipid Effect. Now, red blood cells only undergo anaerobic metabolism. They have no mitochondria. So as a byproduct of this, one of the intermediates of glycolysis is bisphosphoglycerate. This bisphosphoglycerate will bind to the hemoglobin and promote oxygen unloading. An elevated body temperature, like a fever, stimulates bisphosphoglycerate synthesis, as do thyroxine, growth hormone, testosterone, and epinephrine. Thus, all of these hormones promote oxygen unloading in the tissues. The rate of CO2 loading is also adjusted by the varying needs of the tissues. Low levels of oxyhemoglobin enables the blood to transport more CO2. This phenomenon is called the Haldane effect. So oxyhemoglobin does not bind carbon dioxide as well as deoxyhemoglobin. So when there's a high oxygen demand, we also, of course, have a high carbon dioxide generation. And thus, we're allowed to pick up more carbon dioxide because more oxygen is being stripped off the hemoglobin. Deoxyhemoglobin binds more hydrogen ions than oxyhemoglobin. By removing hydrogen ions from the solution, deoxyhemoglobin also shifts the carbonic acid reaction or the bicarbonate buffer reaction to the right. Thus, a high metabolic rate keeps oxyhemoglobin levels relatively low, thus allowing more carbon dioxide to be transported. Pretty cool. I think it's amazing, again, how nature is just so. All of this stuff, is it an accident? Kind of wild. So now the question, we have all these receptors throughout our circulatory system that detect the chemicals and the pressure composition of our blood. How do these gases regulate the respiratory rhythm? The rate and depth of breathing need to be adjusted to maintain the levels of pH at 7.35 to 7.45, the partial pressure of CO2 at about 40 millimeters of mercury, the partial pressure of O2 about 95 millimeters of mercury. These are the set points that the receptors are tuned to. The brainstem respiratory centers receive input, as we've learned, from the central and peripheral chemoreceptors. They monitor the composition of the blood and the cerebral spinal fluid constantly. The most potent stimulus is pH, then CO2, and then less significant is the stimulus of O2. Hydrogen ions have a big impact. Pulmonary ventilation is adjusted to maintain the pH of the brain. As we've seen in many biological systems, if the pH is thrown off, well, so is everything. These central chemoreceptors we learned about in the medulla oblongata produce about 75% of the change that we see in respiration. Hydrogen ions do not cross the blood-brain barrier very easily. So how does this happen? CO2 actually does cross the blood-brain barrier very easily, and it reacts with water to produce carbonic acid. Here, inside the blood-brain barrier, the carbonic acid dissociates into bicarbonate and hydrogen ions. This is the source of hydrogen ions that stimulates the brain. Hydrogen ions are also a potent stimulus to the peripheral chemoreceptors. They produce the remaining 25% of respiratory response in response to the pH change. When we have a blood pH lower than 7.35, we're in a condition of acidosis. If it's higher than 7.45, it's alkalosis. We'll address more on this in a couple of chapters. Hypercapnia is the condition of having higher than 0.43 millimeters of mercury for the partial pressure of CO2. This is the most common cause of acidosis. Hypocapnia is having less than 37 millimeters of mercury for the partial pressure. This is the most common cause of alkalosis. Respiratory acidosis and respiratory alkalosis result from imbalances in pH that occur because of a mismatch between the rate of pulmonary ventilation and the rate of CO2 production. Hyperventilation is a corrective homeostatic response to acidosis. If we blow off CO2 faster than the body produces it, we can push the reaction to the left. We're expiring a lot of CO2, and thus we can draw hydrogen ions out of the solution. This reduces the hydrogen ion and thus reduces the acidity and raises the blood pH back towards normal. Hypoventilation is a corrective response for alkalosis. It allows CO2 to accumulate in the body and shifts the reaction to the right. This raises the hydrogen ion concentration and thus lowers the pH back towards normal. Ketoacidosis is brought about by rapid fat oxidation. It releases acidic ketone bodies. These are the common smell that we get from diabetes mellitus. It induces cosmol respiration, which is hyperventilation. But the hyperventilation cannot remove the ketone bodies. It can blow off CO2, and that reduces the CO2 concentration and thus reduces hydrogen ion concentration and compensates for the acidity of the ketone bodies to some degree. But eventually, we do need to intervene and correct the condition. Carbon dioxide has indirect effects on respiration, primarily through what we've observed with pH and hydrogen ions, because of the bicarbonate buffering system. It has direct effects. During the beginning of exercise, it can directly stimulate the peripheral chemoreceptors and trigger higher ventilation rates more quickly than the central chemoreceptors, simply because the increased carbon dioxide levels get there more quickly. Oxygen has very little effect on respiration. An arterial PO2 significantly affects respiratory rate only if it drops below 60 milliliters of mercury. At low elevations, such a low PO2 seldom occurs, even with prolonged holding of breath. We'll see that a moderate drop in partial pressure of oxygen stimulates the peripheral chemoreceptors, but another effect overrides this. As the level of oxyhemoglobin falls, the hemoglobin binds more hydrogen ions. This raises the blood pH, which inhibits respiration and counteracts the effect of the low partial pressure of oxygen. When we get up to very high altitudes and the partial pressure does fall below 60 millimeters of mercury, the stimulatory effect of hypoxemia on the carotid bodies overrides the inhibitory effect of this pH increase. This produces heavy breathing in people who are not acclimated to high elevation. In the case of long-term hypoxemia, we can lead to a thing called hypoxic drive, in which respiration is driven more by the low partial pressure of oxygen than it is by the partial pressure of CO2 or pH. This occurs in situations such as emphysema and pneumonia, because those are interfering with the alveolar gas exchange. It also will happen in mountaineers who are not acclimated to that level after about 2 or 3 days of duration. But in general, the effects of oxygen have way less of an effect. And as you're all probably aware, our respiration rates and respiration volumes increase during exercise. The cause of this increase is twofold. The brain will send motor commands to the muscles. At the same time as the brain sends motor commands to the muscles to increase their activity, it also sends information to the respiratory centers. So we see an increase in pulmonary ventilation in anticipation of the needs of exercising muscles. There's also a phenomenon where if you're running and you look and see a big hill, the respiration rate and heart rate will increase in anticipation of climbing that big hill. Exercise also stimulates proprioreceptors of the muscles and joints. They transmit excitatory signals to the brainstem and the respiratory centers. This increases breathing because the brain has been informed that the muscles have been told to move or are actually moving. The increase in pulmonary ventilation keeps blood gas values at their normal levels in spite of elevated O2 consumption and CO2 generation by the muscles. There are a number of different respiratory orders and oxygen imbalances. Your text goes into many more of these conditions. Perhaps you're interested in highlighting one of them for your final project. Hypoxia is a deficiency in oxygen tissue, and it's a consequence sometimes of respiratory disease. Hypoxemic hypoxia is a state of low arterial partial pressure of oxygen. It's usually due to inadequate pulmonary gas exchange. Oxygen deficiency at high elevations, impaired ventilation, i.e. drowning, aspiration of a foreign body, respiratory arrest, or degenerative lung diseases can bring on hypoxemic hypoxia. Ischemic hypoxia is when we have inadequate circulation of blood. This can happen as a result of congestive heart failure. Anemic hypoxia is due to the inability of the blood to carry adequate oxygen. Histotoxic hypoxia results from metabolic poisons such as cyanide. These poisons prevent the tissues from using the oxygen, even if plenty of oxygen is being delivered to them. The condition of cyanosis is when we have blue skin. This is a big sign that hypoxia has come on. It is possible to get oxygen toxicity. It results from pure oxygen breathed at 2.5 atmospheres or greater. Now, it's safe to breathe 100% oxygen at 1 atmosphere or below for a few hours, but if we have prolonged consumption of high levels of oxygen, it generates a lot of free radicals. The free radicals are H2O2s or 1/2 O2s, and these destroy enzymes. It's free radicals that cause cancer. Not only do they destroy enzymes, but they destroy DNA and damage nervous tissue. Oxygen toxicity leads to seizures, coma, and death. This oxygen toxicity is why scuba divers breathe a mixture of oxygen and nitrogen rather than pure compressed oxygen. Hyperbaric oxygen was formerly used to treat premature infants for respiratory distress syndrome, but it caused some retinal deterioration and blindness in many infants, and thus the practice has been discontinued because the possibility of oxygen toxicity is just too much of a risk. Now, you've probably all heard of COPD and a number of commercials and treatments for it. That COPD refers to any disorder in which there's long-term obstruction of airflow and a substantial reduction in the pulmonary ventilation. That means any condition where someone's not getting enough oxygen because of issues in the respiratory system. Major COPDs are things like emphysema and chronic bronchitis. Your text goes into a little bit more detail about some of these conditions, but they're usually associated with smoking or breathing some other environmental pollutant. So this ties up our chapter on respiration. It's a lot of information. Spend some time putting together your diagrams and understanding how gases are exchanged between the lungs and the blood, as well as the blood and the tissues, and back again. That's the predominant portion of our diagramming for this week.